Hey everybody, today we are talking about biologics and the types of biologics that are created by biotech companies. Before we start, we're going to get into a quick why is this important to know. Knowing the types of biologics allows you to distinguish the different types of biotech assets from each other, and it gives you a good sense of each of these types of biologics, or what I will call modalities for most of this video, has distinct properties that give it pros and cons. And so it's important to know the pros and cons of each, but we'll kind of just very highly glimpse through what are the types of biologics. So we'll go in order from kind of smallest to largest. So if we're thinking very, very small, I'd say RNA therapeutics or DNA and RNA therapeutics are right there. So what are these? These are therapies that are made out of DNA or RNA. If you guys don't know what DNA and RNA is, real quick, this is known as genetic material. DNA you can think of as genetic material like blueprint. RNA you can think of as genetic material that is like a copy of. And so these two are used. DNA is transcribed into RNA, and then RNA is then translated into a protein product, and proteins are the machinery of the cell. So this is really quick zooming through a lot of different concepts in biology, but DNA and RNA therapeutics are probably the smallest. These are often used, and they're really up and coming and really hot right now. But the idea is that you insert the DNA or RNA into a cell, and the cell goes through that process, as I just mentioned. So it transcribes DNA into RNA, and then RNA into a protein product. It could be an enzyme. It could be a receptor, many whole host of things, but basically the protein acts like a little machine and has a purpose. So this machine, based on its purpose, will then carry out some type of function in the cell. So this is, again, just a really high-level way of thinking about it. That's how, so DNA, RNA therapeutics. Again, really, really popular right now, and there's many applications to it. One of the pros to this would be that if it's a protein that needs to continually be expressed or produced within the cell, if this machine, if you want to think of it as a machine, if this machine needs to be constantly made within the cell, then this would be a great DNA and RNA DNA therapeutic would be a great solution, and RNA therapeutic potentially if you only want it for a short period of time, the duration. Cons to this is it's really hard to get DNA into a cell. Moving forward, from there, after DNA, RNA therapeutics, another type of biologic or modality would be peptide therapeutics. Peptides, yeah. So peptides are really small proteins. So we talked about DNA and RNA. Then we go to peptides are the product of these, and they can be the order of magnitude difference from DNA and RNA, I'd say, could be 10, 100x, 10x probably. But they could be like messengers, molecules. So the production of this particular molecule messages the rest of the cell to produce something or to turn off something, to upregulate gene expression. When we say gene expression, we mean upregulate the production of a protein product or downregulate the protein product itself. So you may want to have higher levels of protein or lower levels of protein based on whatever the disease or condition is and how the molecules flow and create life, if you will. From proteins or peptide therapeutics, again, it talks about they're pretty small. Advantages to these are they're small. They don't necessarily it's easy to get into the body and into particular cell types. Disadvantages, though, could be that since they are so small, that they could be attaching to a bunch of different things and really make an issue in terms of side effects. So again, these are I'm just running through these really quickly. There's a lot more detail to this, but this is just very high level. Another piece would be antibodies. These are a pretty big modality and one where many drugs in biotech and pharmaceuticals are being developed. They could be monoclonal, they could be bispecifics, which bispecifics are pretty big right now in terms of an idea. And so these would be often useful for working in conjunction with the immune system. It could be that they are great at targeting and turning off or digesting particular molecules, like so a signaling molecule, for example. If an antibody attaches to it, then your immune system goes and eats it and kills it. So that whatever that gene product might be, you might have an antibody against it, and it would digest it. Oftentimes, these are used in cancer because you might want to shut down a particular signaling molecule, or you might want to attach to a cell itself. By attaching the antibody to the cell, the macrophages, the immune system, the white blood cells will go and eat it. So those are antibodies, again, at a high level. There's antibody drug conjugates, where it's, again, like the Y shape of an antibody, but then on the bottom piece, there may be a cytotoxic molecule or something that kills cells. This is often used in cancer. So it's just spurring my mind going wild. But it may have some type of small molecule drug that can kill the cell. So it's kind of used as like a homing device for the payload, if you will. So those are ADCs, antibody drug conjugates. From there, you may have, cell therapies are a lot larger as well. I would say cell therapies are probably your next category of size, if you will, or modality of biologics. So these could be anything from stem cells to CAR T cells, particularly for cancer, or immunotherapy, increasingly so probably for autoimmune disease as well. You may see some just any like white blood cell, NK cell, all that fun stuff. You might also see just cardiac cells or muscle tissue or muscle cells, skin cells for grafts. That's even larger than cell therapies. That would be tissue therapies. But again, pros of cell therapies is that if you need a machine to continually produce a particular type of gene product or protein or peptide, this would be a great modality to go about doing that, because it provides some structure for this machinery. It also might be useful for anything where there's structure itself. So if there's an issue with structure of a vein, an artery, or some such thing, or even just tissue, connective tissue. This would even include skin, cartilage, etc. Cells are great because they provide structure. So they have space with which they take up, and so cells are great that way. Moving forward, we have tissue. Tissue, yeah, living tissue or tissue approaches would be the last type of. You could even go probably larger than that, but we'll end it at tissue/organ. So imagine that you regenerate muscle tissue or skin tissue. If you have like burns, third-degree burns, you might want to have like a patch of skin that you could just place on. It anchors to your skin and it becomes your own. Muscle tissue, same thing. Cartilage should be a great example of that. Heart tissue, what have you. So tissue itself. And then organs. So imagine growing a whole heart itself, or growing a whole liver, or growing a whole kidney, and being able to grow it outside into a lab and then implant it into the body. Would be revolutionary. Anyways, that's kind of the different types of biologics. When we say biologics, we're including everything except small molecules. So again, this all has to do with drugs. What are the size of drugs themselves? Recognizing that there's pros and cons to each of these modalities, and knowing that we went through very, very quickly most of these just to really glimpse through and kind of talk about what are some of the differences and what are some of the really popular ones. And then lastly, reiterating again why this is important, to get an understanding that there are differences to these. And so by using one or the other to deliver a drug or to enact on the biological system of a person, benefits and causes for concern. So things to remember or to be aware of. And I hope you enjoyed this video. Let me know if you guys have any questions. Throw it in the comment section. And with that, I leave you guys. Peace.